secondary structure has two examples of ordered secondary structure. The first example of ordered secondary structure is alpha helices, so forming a sort of spiral staircase. And there are hydrogen bonds, which are shown here by dotted lines, between R groups in different areas of the primary sequence that have come together to hold this alpha helical structure together. So you can see if we follow the backbone of the staircase up, that there's different R groups in different parts of the primary structure. They're not adjacent to one another, that are forming hydrogen bonds, and it's those hydrogen bonds that are holding together this helical structure. This is a very, very strong arrangement. For example, human hair is made up of alpha helices. The alpha helices are in a protein that's called keratin. As we'll see in a minute, there's a tertiary structure, so we'll have three alpha helices twisted together in a cable. This is what gives hair its strength and flexibility. And if we want that hair to curl, or if we want to straighten that hair, what we can do is break disulfide bonds between the different keratin strands and then reform them in a different way. And in that way, we can either curl or straighten hair. That's called a permanent or a straightening process. Because it uses sulfur-containing compounds, the process smells very bad, and it's chemically very hard on hair to do this to it. But just sort of a little gee whiz fact showing you that I can use the factor label method to calculate all kinds of different things. I was curious at how fast it is that we make these alpha helices when we're making hair. So if hair grows at a rate of about a ½ inch a month, which it does, we can use that information along with some information about the dimensions of the keratin molecule to figure out that we're making about 26 amino acids per second in the primary structure, or 7 turns of the alpha helix a second in the secondary structure of keratin as it's being made. So as I'm sleeping, as I'm going about my everyday activities, I'm spinning out about 7 turns of this alpha helix every second that I'm alive as my hair is growing. The second example of secondary structure, so we have 2 examples of secondary structure, and the second one of those is what's called a beta-pleated sheet. A beta-pleated sheet, like an alpha helix, is also held together by hydrogen bonds. So once again, we have non-adjacent amino acids that don't follow each other in the primary sequence, that have come together and through their R groups interact with one another to form a flat sheet-like structure, or actually a pleated sheet looking like pleats in a skirt, for example. When we illustrate these protein structures, we show alpha helices as coils. That's not too surprising. What may be a little surprising is we show the beta-pleated sheets as flat arrows. And where in areas of the protein where we don't have secondary structure, we just show them as threads. An example of this from human medicine is shown where we have a normal amyloid protein which normally has 4 alpha helical coils in it with some thread-like secondary structure that's unorganized in between. But when it undergoes pathological changes, a couple of those alpha helices turn into 2 more beta-pleated sheets, and so we end up with beta-pleated sheet areas, and this gums up in some way that we don't fully understand. This gums up the works of neurons and causes them to not work very well in Alzheimer's disease. Next, we'll look at tertiary structure, and that's how we put together these alpha helices, the beta-pleated sheets, and the unorganized secondary structures into 3 dimensions. So just remember that 3 dimensions has to do with tertiary structure. For example, insulin consists of 2 chains, shown here as chain A and chain B. Each of those chains consists of a primary and a secondary structure, and when we put those 2 chains together into a three-dimensional structure that makes a functional insulin molecule, then we call that tertiary structure. The kinds of interactions that lead to the formation of tertiary structures are ionic bonds. Ionic bonds are simply charges. So we have one R group with a positive charge and one R group with a negative charge that are attracted to one another. We have hydrophobic interactions. Those are simply molecules that are expelling water, and so they're liking to hang out with each other. We have van der Waals interactions. Those are very, very poorly understood, but the basic idea is that we think that there are different shapes of molecules that like to hang out with other shapes of molecules. We have disulfide bridges, as we talked about earlier, between cysteine R groups. And we have hydrogen bonds. And notice that the things holding together these tertiary structures, just like we saw for secondary structure, the things that hold together these tertiary structures are R groups. And those R groups don't necessarily, and probably we don't even want them to come from adjacent amino acids. These are distant amino acids that have R groups that begin to interact with one another and form the tertiary structure of these proteins. Let's look at another concrete example of a tertiary structure. This is the triple helix that we find in keratin fibers. Notice that each one of these protein strands is made up of a long continuous alpha helix. And when we take three of these alpha helices together, 1, 2, 3, and twist them into a cable, so now just like we take wires in a cable and we twist them together to make them even stronger than the individual wires, then we can twist these three alpha helices together for increased strength. And that's what we do in a keratin fiber. Now just to review once again, there's 4 levels of protein structure: primary, secondary, tertiary, and quaternary. We're up to quaternary structure now. So in Objective 30, we'll look at the quaternary structure of proteins. A good example of quaternary structure is the hemoglobin molecule. This is going to be the main oxygen-carrying molecule for the body. In order to carry oxygen, we want to use iron-containing groups called heme groups. These heme groups are nestled in polypeptides that are called alpha globins or beta globins. The alpha globins are identical to one another. They don't look at this diagram, but one of these is just turned relative to the other, so this occurs by rotation as we're putting this together in a different way. So we have 2 identical alpha globin polypeptides. We have 2 identical beta globin polypeptides. We put 2 alpha globins and 2 beta globins together, each with its own heme group. We have 4 heme groups able to carry 4 oxygen molecules. And this hemoglobin results from the primary structure of the individual amino acids in the individual strands, alpha helices and beta pleated sheets, forming secondary structures. Those alpha helices, beta pleated sheets, and unorganized secondary structures are all put together into a 3-dimensional tertiary structure, an alpha globin polypeptide or a beta globin polypeptide. And then we put the alpha globin polypeptides and the beta globin polypeptides into a quaternary structure, 4 subunits, making up a complete hemoglobin molecule. The R groups are critically important here, and if we change the function of an R group, if we change the structure, of an R group by changing the amino acid that we find there, then we can completely mess up this process, as we'll see later on in the disease sickle cell anemia. So this slide takes you through primary, secondary, tertiary, and quaternary structures step by step. The primary structure of a protein, as I said earlier, is its sequence of amino acids forming the polypeptide chains. The secondary structure is either coiling in the case of alpha helices or folding in the case of beta-pleated sheets of the polypeptide chains. Tertiary structure is the three-dimensional shape of the polypeptide. So that's the areas of the primary structure that have formed secondary structures, alpha helices, beta-pleated sheets, and then just some loosey-goosey structures. And how we fold together those alpha helices and beta-pleated sheets and loosey-goosey structures is what we call tertiary structure, which gives a protein three dimensions. And then quaternary structures, when we take those three-dimensional protein chains and we put them together into one big structure. It's called quaternary structure. This is the same example as seen earlier of the hemoglobin molecule. The abnormal formation of a protein, the abnormal formation of quaternary structure in the hemoglobin protein in sickle cell anemia is shown in the attached movie.